The overall goal of this procedure is to fabricate supported biomembranes by the solvent-assisted lipid bilayer method.
This method can help answer key questions in the membrane biology field, such as understanding the fundamental properties of lipids and membrane proteins, as well as to enable applications such as drug discoveries and molecular diagnostics. The main advantage of this technique is that it can fabricate support lipid bilayer in many cases, which are not possible with the conventional method. The procedure is also easy to use and require minimal sample preparations.
The implications of this technique extend toward therapy and diagnosis because it allows studying important drug targets such as membrane proteins.
The method can also be used to fabricate biocompatible substrates for material science applications.
We first had the idea for this method when we discussed the difficulty of conventional membrane fabrication methods. Demonstrating the procedure will be Minchul and Saziye, two graduate students from our laboratory.
Prepare a lipid stock solution of 10 mg per milliliter DOPC lipid by dissolving the lipid powder in isopropanol solution. Then, dissolve rhodamine-labeled DOPE lipid powder in isopropanol solution to obtain a lipid stock solution of 1 mg per milliliter. Dilute and mix the stock solutions in isopropanol to prepare the desired lipid mixture at the final concentration. For fluorescence microscopy and fluorescence recovery after